Let's talk about the types of ELISA. ELISA is basically based on a principle of epitope interaction. Epitope is a segment or sequence of amino acids in a linear or three-dimensional protein that binds with the antibody or antibody binding site. This sequence or the epitope are variable. One polypeptide chain can have more than one epitope, which can bind to different antibodies. When we think about the types of ELISA, we can categorize them broadly in four types: direct ELISA, indirect ELISA, sandwich and competition और inhibition ELISA. अब हम देखेंगे इनमें डिफरेंस क्या है. डायरेक्ट ELISA एक सिंपल और फास्टर मेथड है टू डिटरमिन द प्रेजेंस ऑफ एनी एंटीजन. इस मेथड में हमारे पास जो मल्टी वेल प्लेट है उसकी बेस को कोट किया जाता है with a known antigen or with an antigen. Then this antigen is detected by an antibody, and this antibody is conjugated with an enzyme. अब इसमें होता क्या है, हमारे पास multi-well plate है, we add the sample on it and incubate it for some time. The antigen binds with the base of the well, then we add the specific antibody, and this antibody is coupled or conjugated with the horseradish peroxidase enzyme. We add the substrate and we get the signal if the conjugation is there. This method has two advantages. It is a faster method and it is a less prone method for errors because fewer or very few number of steps are involved to detect this particular method, to get the results from this particular method. Indirect ELISA slightly differs from the direct ELISA. In this method, the basic principle is the same, that the well is coated with the antigen or with the sample, and then we add primary antibody. This primary antibody is meant for the protein or antigen of our interest, जिसको हम डिटेक्ट करना चाहते हैं या जिसके साथ हम इस एंटीबॉडी को बाइंड करना चाहते हैं. प्राइमरी एंटीबॉडी जो है, दैट इज नॉट कपल्ड विद एनी एंजाइम, रादर देयर इज अ सेकेंडरी एंटीबॉडी which can bind with this primary antibody, and this secondary antibody is coupled with the enzyme so that we can detect the presence of this secondary antibody. So wherever the primary antibody is bound with the antigen, there we will see the binding of the secondary antibody, and it is possible that we can detect the antigen by indirect ELISA. This method also have advantages over the direct ELISA because it involves more than one antibody and it has increased sensitivity. This method also have some flexibility because secondary antibody can be used with different types of primary antibodies, and at the same time this becomes cost effective, that we don't have to purchase every time new secondary antibody. We can use the same secondary antibody. The only thing we need to arrange or we need to purchase is the primary antibody, which is specific for a specific antigen.